This is the plants chapter, and we're going to start off the plants chapter with a seed dissection. We are going to be dissecting a lima bean. This is a seed. We're doing this one because it's very big and it'll be easier to see. The way seeds are classified, they are either monocots or dicots. The seed that we're going to dissect today is a dicot. Di means two. The other kind is a monocot. Mono means one. One cotyledon. When a monocot grows, it's going to be one primary leaf. So examples, corn. Corn is an example of a monocot, as is bamboo, onions, tulips, and coconuts. Dicots have two cotyledons, right? And then when they start to grow, you will see that there are two leaves. Examples, apples are dicots. Maple trees are dicots. Beans are dicots. And daisies are also dicots. So I bought large lima beans, and the first thing I'm going to show you is the first part of the seed that we're going to talk about is the seed coat. And that's exactly what you are seeing right now. The only part of the seed you are seeing is the seed coat. So what I want to show you is the purpose of the seed coat is to protect the seed. So there's a little baby inside of here and it needs protecting. So if we go ahead and go down here, okay, the seed coat, oh, and I have this really super fancy tool provided by the school district that I don't know if you can find these anywhere, but yeah, can you see it? Can you see it okay? It's cool because it's almost like a little pocket knife because it's all folded, and to use it, you just unfold it like that. This is called a seed coat extractor thingy. What I'm going to do is I'm going to use this fancy seed dissection tool, and I'm going to try to get the seed coat off of the seed. So in class, we would do this, and I would give each of you one of these tools, but I would make you put a deposit down on them because they're so expensive, and then when you return it, I give you your deposit back. What we do is we try to figure out how to get through this seed coat, and I let you all try this. And you can scratch at it. There's usually a little notch, maybe another one. Let me see if I can find one. Oh, maybe this one. Can you zoom in and see that little notch right there? So sometimes you can wedge it in right there. And so usually I let you do this in class and I give you about 10 minutes. And sometimes people like slice their fingers with the fancy tool. And that's pretty exciting. And other times it takes a while till somebody finally cracks a little piece. Oh, did you see that little tiny piece came off there? Yeah, a little bit of the seed coat, yeah. So sometimes it takes a while. And then I reveal to you that this is totally unnecessary. The whole purpose of this is to show you how hard the seed coat is and how protective it is of the seed. So you can see not only is it hard to do, but it also makes a big mess because by the time we're done, there's little pieces of seed everywhere. All right, so that is very time consuming in class. I would just let you keep hacking at it until, you know, at least one group, one table had it open so you could look at it, right? What you'll notice is that, you know, I've kind of chipped some pieces off and there's not a whole lot going on there. But the reason why this is called a dicot, two cotyledons, it breaks in half. And the inside the seed coat, each one of those is called cotyledon. And that's the food source, the energy source for the growing plant. What you can't see very well here because it's all dried up is the embryo of the little baby plant. So it's in there, it's like there, but don't worry because I'm going to show you then what I reveal to you in class is the second part. What I have here are some seeds that have been soaking in water for about an hour. The purpose of this, and it's only been soaking for an hour, so, is to show you that the seed coat, as hard as it is, because the seed is dormant and it needs protection, the one thing that it needs to come back to life, or to, I don't know, to grow, before it can grow, it needs to get wet and it needs to shed this seed coat. So if you look at it now, see how easily this seed coat is peeling off? So when it's wet, it starts to break down, and look at that. So if I would have just given you the wet seeds to begin with, that would have been so much easier. But one thing you'll notice compared to one of the beans that hasn't soaked is that the seed coat is really smooth on this, and on this one, see how it's all wrinkly? So as it starts to soften up, it gets all wrinkly, and then it's super easy just to peel off the seed coat. There we go. Yeah, so that right there, that is the embryo. So the 3 parts of the seed that you need to know: the seed coat. The seed coat is the hard shell that's protecting the seed. When it gets wet, it's going to eventually soften and come off. The cotyledon is the energy. This is how the plant gets its energy to grow. So it's like food. That's the source of energy it needs to grow when it's in the ground. First, the water gets into it, it softens the seed coat, and then the plant starts to grow using the cotyledon for energy. The little baby plant here, sometimes you can see it has a little leaves on it even, right? So that's what's going to turn into the plant using this source of food. Now, eventually, it's going to break out of the soil, and it's going to have stems that are going to reach up, or shoots, right? So it's going to come out of the soil with shoots that will then have leaves. And by the time it gets there, this cotyledon will be all used up. There won't be that anymore because the plant has used it. So as the plant grows, this shrinks, it's used up all that food, and by the time it's out of the soil, it will then use its leaves for energy through photosynthesis, right? So that's what we'll be learning about. And so in the order that what we're going to be doing in this chapter, we're going to start with seeds, and then we're going to learn about roots and shoots. Shoots are like the trunk of the tree, the branches, roots, shoots, and leaves and flowers. And then it cycles right back to seeds again. So it's the whole life cycle of the plant. So the next lab you're going to see is germinating seeds, and that's what we will be doing next.